Hello, my name is Jackson Shelley, and as you can see, today is 04/02/23. Today we're going to be discussing the movement, of macromolecules. First up, we have progesterone. Progesterone is an essential hormone that is involved in many reproductive processes, including menstruation and conception. The synthesis of progesterone begins with the uptake of cholesterol from the bloodstream into the cells of the adrenal cortex or ovaries. So after it has been uptaken from the bloodstream, it is transported to the mitochondria for the conversion into pregnenolone by the cholesterol side chain cleavage enzyme CYP11A1. Pregnenolone is the precursor to all steroid hormones, and as we can see in the next step, progesterone is synthesized from pregnenolone by two enzymes, and those are 3-beta-hydroxysteroid dehydrogenase and delta-4,5 isomerase. These are both located in the smooth endoplasmic reticulum of the cell. Next, we have the transport from the smooth endoplasmic reticulum to the Golgi apparatus. So after the synthesis of progesterone, it needs to be transported to the smooth endoplasmic reticulum to the Golgi apparatus. This is accomplished by a carrier protein that moves the progesterone from the smooth endoplasmic reticulum to the cytoplasmic surface of the Golgi. Within the Golgi apparatus, the progesterone is modified by enzymes such as sulfotransferase, which adds a sulfate group to the hormone, making it more water-soluble. Once it is modified, the progesterone is packaged into secretory vesicles within the Golgi apparatus. These vesicles are then transported into the cell membrane. Then we have the secretion of progesterone. So those secretory vesicles that were just formed containing progesterone fuse with the cell membrane, releasing the hormone into the bloodstream. From there, the progesterone travels to its target tissues where it can exert its biological effects. The fate of progesterone: once it is secreted, progesterone can act on its target tissues, such as the uterus, mammary glands, to regulate the reproductive processes such as menstrual cycles and pregnancy. As you can see, here's an overview. Next, we have insulin synthesis. Insulin is a hormone that is essential for controlling blood glucose levels. Its synthesis starts in the ribosomes of the pancreatic beta cells, where a larger precursor protein, pre-proinsulin, is produced. Pre-proinsulin consists of a signal sequence, the insulin A chain, a linking peptide, otherwise known as a C chain, and the insulin B chain. So, after that is formed, as pre-proinsulin is translocated to the endoplasmic reticulum, the signal sequence is cleaved by signal peptidase, resulting in proinsulin. So, then we have the disulfide bond formation inside of the lumen of the endoplasmic reticulum. Proinsulin will fold, and disulfide bonds will be formed between cysteine residues, stabilizing the protein structure. Proinsulin is then transported from the endoplasmic reticulum to the Golgi apparatus and transport vesicles. In the Golgi apparatus, proinsulin will be modified further. Proteases will cleave the C-peptide, separating insulin's A and B chains from each other, removing the C-peptide. The resulting 2-chain molecule is now insulin. The Golgi also adds carbohydrate groups to insulin, which will be essential for its proper folding and function. After that, mature insulin is then sorted into secretory vesicles, which will be sorted in the cytoplasm of these beta cells until it is time for release. Upon stimulation by glucose or other secretogues, the secretory vesicles containing insulin will fuse with the plasma membrane, or otherwise known as the cell membrane, releasing the insulin into the extracellular environment. The fate of the insulin: from there, the insulin will enter the bloodstream, traveling to its target tissues where it can regulate blood glucose uptake and utilization. Here, as you can see, we have another overview. After that, we have actin synthesis, which is our intracellular protein. Actin is a protein that is essential for many cellular functions, such as cell shape, movement, and division. Actin synthesis starts with DNA transcription to mRNA which is then translated by ribosomes. These ribosomes read the mRNA sequence and build the primary structure of actin protein by adding amino acids in the proper order. So, after that, it is transferred to the rough endoplasmic reticulum, and the primary structure of actin undergoes the process called post-translational modification. This includes the removal of the initiator methionine, folding of the protein, and the addition of carbohydrates in a process called glycosylation. After that, we have smooth endoplasmic reticulum transport. After post-translational modifications, the actin protein is transported from the rough ER to the smooth ER in transport vesicles. The smooth ER is another network of interconnected membrane sacs, but without ribosomes. In the Golgi apparatus, the actin protein will be further modified by the addition of a phosphate group. This process is called phosphorylation. The Golgi also will sort and package actin into secretory vesicles. After that, the secretory vesicles containing actin are going to be transported to the cell membrane, and from there, the secretory vesicles containing the actin will fuse with the cell membrane, releasing the actin protein outside the cell. The actin protein can then incorporate itself into the cell cytoskeleton or other structures where it plays a crucial role in cell shape, movement, and division. And here you can see we have another overview. And here's our references. Thank you so much.